there we go, and make these recordings available through the MMV webpage, which is find it through mmv.org, and it's on this pull down here. And there are also some guides to free tools. If you haven't come across that page before, it's worth a look. And you can download, for example, information and downloads for the PK tool, which Gav will mention, and DataWarrior, which Isabelle will cover. So please let us know what you think. This is something of an experiment. So tell us what you think, what you'd like to hear more about, and also whether you can volunteer something. So I will. I have one quick poll question to ask because it would be interesting to know just a little bit about the audience today. So hopefully you will see a quick question pop up in front of you. If you can click on that, and I will share the results with you very soon. Okay, all done? Pretty much. Okay, from the 88% of you that voted, these are the results. Okay, so I'd like to move on now to introduce Gav Whitlock, who's going to give us our first presentation. I think I have to hide that poll. Gav, are you able to share your presentation? Gav, I think you may be muted. Gav?
Mark. Yeah, Mark, you muted me for some reason.
Sorry about that.
Clearly you didn't want me to talk. Okay, so Mark, have you given me presenter rights?
I thought I had.
Well, can you see my screen?
No, it says waiting to view Gav Whitlock's screen here. Button on the top left. This worked when we dialed in, folks. There we go.
Okay, yeah.
Excellent.
See it now?
Yep.
Right. Let me just put it onto SlideSoja. Thanks, Mark. Thanks for organising these meetings. I'm really looking forward to taking you through what we've got, and be very, be great to get questions either at the end of the presentation or by email afterwards if we run out of time. So what I want to take you through is just some of the ways that we've been using PK tools, and that's either using Excel or the PK tool developed by Xenologic, and how we've used those tools in the optimization of a series of compounds for the treatment of Leishmaniasis. Okay, so this is what I'm going to go through briefly today. A little bit of project background just to give you some context of the chemical series and where the project started, etc., etc. a little bit of brief summary of some of the early SAR that we've seen. Some of that's now published, and I'll give you the reference for that particular article, and then really jump into the meat of the presentation, really, in terms of how we're using in vivo ADME data, and in conjunction with an Excel tool looking at PK superposition to try not only pick out the best compounds, but then what doses do we take those compounds into in vivo efficacy studies at. Touch a little bit on some thoughts on free versus total drug levels and how that can impact on in vivo efficacy. And then from there go on to talk about the PK tool itself and how we've been using that to not only understand what our human PK and dose projections might be, but also thinking about safety margins over some off-target activity, and then wrap up with a quick summary at the end. Okay. So a little bit of project background. So this is a project that is has multiple different collaborators involved in it. The leadership of this project is through DNDi. Ourselves at Sandexis work closely with some colleagues at Takeda with respect to compound design, and we also look to them for some other expertise in terms of safety and tox when they're able to provide that. This project is funded under the auspices of the GHIT Fund, who have generously supplied funding for this project for 18 months to get us to candidate selection. A couple of different centres for parapathology. The University of Antwerp, Louis Maze's group there, who will focus on L-in phantom screening, both in vitro and in vivo in the hamster model, and then we complement that as needed, looking at Donovani screening in the mouse at the London School. And certainly not forgetting our involvement with Wuxi here, who do all of the synthesis and all of the DMPK work, both in vitro and in vivo. So quite a complex project setup, but we all work really closely together to try and make this work as smoothly as we can. Okay, so as I said in the title, this project's really targeting an oral small molecule therapy for Leishmaniasis, primarily visceral Leishmaniasis, but we're being opportunistic here and looking at compounds in vitro and in vivo screens for cutaneous Leishmaniasis as well. In terms of a headline product profile, we're looking for a once-a-day or twice-a-day oral dose compound for ideally no more than 10 days of dosing. And what I've got on this slide is just a very quick snapshot of the early screening sequence that we use in terms of design, synthesis, screening loop. So ourselves and Takeda at the top both work very closely together with respect to compound design. Wuxi then take over with respect to synthesis, and then in parallel we screen compounds in Leishmania screening in primary mouse macrophages at LMPH in Antwerp, and in parallel to that the primary mouse macrophage cytotoxicity to make sure that any activity we see in the parasite screening is, we think, on target and not related to cytotox. And then we have a human MRC5 cytotox counter screen as well. If compounds pass certain criteria with respect to those screens, then we move on to a whole bunch of in vitro ADME screening, human, mouse, hamster liver microsomal stability, solubility, log-D, MDCK permeability. And after we've generated a lot of that data, we'll make a decision as to whether we progress compounds to hamster PK, so that's oral PK, a single dose of 50 mg per kg, and then really it's the analysis of all of this information to determine whether the compound goes forward into the hamster early curative model of VL, typically at 50 mg per kg twice a day, but then you'll see some examples where we've actually changed that dose, and then we'll decide whether the compound's active or not, and then if it is, then you'll see later on when we talk about the PK tool how we've taken some of those compounds forward. So for the majority of the people on this call who are chemists, here's the first structure. So this is the original hit that this program started with, which came from a screening a diverse set of compounds from the Pfizer file, and Mark himself was really instrumental in making sure that this happened. As you can see from the data on this slide, it's not a particularly active compound. So against L-donovani and L-infantum, we're looking at somewhere between 10 and 40 micromolar, if we're being reasonably generous. So a weak hit, but I think an interesting looking structure, nice, low molecular weight, not particularly lipophilic, and you know, we really felt that this was a good starting point to start a hit to lead program. And that hit to lead program lasted for probably around 2 years, and we were able to deliver a range of very potent compounds. So we really significantly improved the potency of that compound and also its in vitro ADME properties. And the early SAR is really described in this JMed Chem article, which came out at the end of last year. And that really took us to this compound here on the left, this 4-fluorophenol piperazine urea with this pyridyl aminopyrazole on the left. So this compound is significantly more active than where we started from with the lead, so we're now around 1 to 2 micromolar in vitro against infantum and donovani with clean window over cytotox, and certainly at this point good enough stability in human and hamster liver microsomes. And the hamster is particularly important because that's the model that we're really looking to do efficacy studies in. And when we took that compound into a hamster efficacy study at 50 mg per kg, BID, so that's twice a day for 5 days, we saw really very high levels of reduction in parasite load. So just to put some of these numbers into context, we would say anything above 95% is very, very good. We use miltefosine as the standard in that model, and that typically comes out around the 95% range. So we were, really delighted to see that data, and we felt that was a positive in vivo proof of concept for that series. We generated that data a while ago now, and since then we've been working hard just to optimize this a little bit further. And I'm not able to show the specific structures of the lead compounds right now. I've summarized the general properties of those lead compounds here. So we've improved potency. Again, I'll put most potent compounds above less than 100 nanomolar against both infantum and donovani. Still very good against cytotox. We've improved human liver microsome stability, which will obviously be important for human PK, and also hamster, which means that we can hopefully start dialing the dose down a little bit. And when we look in a hamster efficacy study, we can now hit greater than 99% reduction in parasite load against all of the 3 major organs that we're interested in those studies. So that's all I was going to say about the project background. I guess when we started this, we were thinking hard about, well, what's the best way to try and understand the relationship between dose and exposure and in vivo effect. And I'm sure everyone that's listening to this will appreciate that it's really a very challenging thing to do, to understand what the real drug levels are at the site of action inside the parasite. And so we chose to really be quite pragmatic about this and think about, well, what's the best surrogate that we can use to allow us to set reasonable doses and then make decisions on whether the compound or series should go forward or not. So, for example, if we see low efficacy, then we need to be confident that we've tested the compound at the right dose to give the right exposure, and if it doesn't work, then we know we can walk away from that template, understanding that we've run the right experiment. Counter to that, if we see very, very good efficacy, then I guess we need to think about, well, what's the right parameter in terms of drug exposure to use when we're going to go forward and start what our human dose might be. So we've chosen to think about drug levels in plasma and aim to achieve exposure in the animal model within the same range as our in vitro potency. We then get onto a discussion about, well, are free drug levels or total drug levels more relevant? And just to be clear what I mean by that, so free drug we would define as the concentration of drug in plasma after you've corrected for plasma protein binding. I've just given a simple example at the bottom that highlights that. And beyond that, something that we asked ourselves quite early on is, could you assume that there was sufficient plasma in the in vitro assay to think that your in vitro potency was essentially a bound-up potency value? So can we measure protein binding in the in vitro assay in a similar way to we can measure it in plasma? And so we did that, and that's on the next slide. So across a bunch of compounds, what we've got here is on the y-axis, we've got the fraction and bound of the compound in the assay media, and by fraction and bound I mean the percentage of compound that's free and not bound to plasma proteins, and the way you measure that is you take the plasma protein binding away from 100 to give you fraction and bound. So, for example, if plasma protein binding is 95%, fraction and bound is 5%. So on this plot we've got assay media binding on the y-axis. So this is the assay media used for the L-infantum amastigout assay, and on the x-axis we've got hamster plasma protein binding, so fraction and bound in hamster plasma. And I guess what you'll be able to see from this is the compounds are much freer in the assay media than they are in hamster plasma. Typically, by a 5 to 20-fold higher binding in plasma proteins rather than binding in the assay. So what we took from this is that if we're measuring drug levels from an in vivo PK study, and we're trying to correlate that with in vitro potency, we should probably be correcting the drug levels for plasma protein binding, but we shouldn't need to correct the IC50s from the in vitro assay, because actually there's very little compound bound up. What I would say is that when we get compounds with 50% or less fraction and bound, then we do tend to correct those, but the vast majority of compounds that we've looked at in this series are essentially completely free in the assay media. So the in vitro IC50 that we're measuring is really the free IC50. So when we've generated all of this data, we've got good potency, good cytotox, sufficient stability in hamster liver microsomes and obviously human liver microsomes as well, and we know something about the hamster plasma protein binding and the binding in the assay media. We'll start generating some PK data, and typically we do this in an oral PK study in hamster at 50 mg per kg, single dose. And then what we want to try and do is obviously look at that exposure relative to the IC50, and then think about, well, is free levels or total levels more relevant? And the way we've done that is just to use a simple PK superposition template in Microsoft Excel that we built, and then this allows us to look at a number of different doses and look at free exposure and total exposure, and then make some judgments as to what doses, if any, to test a compound. One of the decisions that might come out of this is we decide not to take it forward. And to try and put this into context, I've got a worked example on the next few slides. I can't show the structure of this particular compound at the moment, but so I've just called it compound 1. And the all dose PK from a 50 mg per kg study in hamster shown on the left, and what I've also done is map on the L-infantum IC50 onto that. So you can see that the exposure is very good relative to the L-infantum IC50 at that dose. So what we've then done is look at modeling that data in Excel across 5 days. We've had to make an assumption here if we're going to look at different doses, and that's that we're going to assume that PK is linear across the dose range that we'll be looking at. So then we can look at how changing the dose changes exposure, both in a total sense and a free sense. So what I've got here is taking that 50 mg per kg single dose data. We've modeled that over 5 days, looking at three different potential doses: 25 mg per kg BID, 12.5, and 6.25. And again, I've mapped on the L-infantum IC50 in this case. And as you can see, based on these plots, we would certainly have expected, or would expect, pretty good levels of efficacy in the in vivo model, because we're well above our in vitro IC50 for the vast majority of the dosing period. An alternative way to look at it then is to look at actually, well, what are the free drug levels? So what happens when we correct our total plasma levels for plasma protein binding, and then we correct the IC50 for assay binding, because this compound's around 50% bound in the assay. And we see quite a different picture. So at 25 mg per kg, we see exposure within a couple of fold of the IC50, again for the majority of the dosing period, probably for 16 out of each 24-hour period, we're within about 2-fold of the IC50. Not quite so good at 12.5 mg per kg, we're only just spiking up to the IC50 each each day, and then a lower dose of 6.25 were a good 2 or 3-fold off the IC50. And that's then reflected in the in vivo efficacy outcomes that we see. So 25 mg per kg PID, we see very high levels of efficacy, up to approaching 99% across all 3 organs. In particular, bone marrow is a very difficult organ to get inhibition up to that kind of level. And as you can see, as we go down the dose, then the bone marrow starts to drop off really quite dramatically just by changing the dose 2-fold, and then nearly all 3 organs are starting to drop off at the lower dose. So, you know, this is certainly not the only experiment that we've done using this Excel tool. We use this quite routinely now, looking at total exposure and free exposure. And we've looked at it not only in this series, but another completely unrelated series, which we think is working through a different mechanism of action. And so far, I think we've got reasonably broad agreement across both of these templates that if we can target free exposure around or above the in vitro IC50 for most of the dosing interval, that's going to give us the best chance of really hitting the maximum efficacy up in the greater than 95% range. Now, that's not to say that this kind of relationship will hold true for every series, but it's certainly an observation that we found, and certainly using the tool in Excel has been extremely helpful to help us understand that. Now, one thing I would say is that as part of this, we've taken single dose data at 50 mg per kg, and then we've modeled that across a bunch of different doses out for 5 days. So one of the things that we wanted to do was actually just double check that if we measured 5-day PK data, it would correlate reasonably well with the models. So that's what we've got on this slide, where the light blue plot here, so this is x-axis is hours, and y-axis is nanogram concentration in nanograms per mil. And in the light blue, we've got our modeled PK data over 5 days. And what we've done here is measure plasma exposure on day 1, and then keep dosing, and then measure plasma exposure again on day 5. And certainly to our mind, the model data and the measured data really look in very good agreement. That the AUC, the calculated AUC and the measured AUC are really very close to each other. So I think this gives us some confidence that we can use this approach of taking single dose PK data and then modeling it out for 5 days. And certainly the Excel tool, you know, we think is fit for purpose and has been very helpful. Okay, so once we've done all of that, and we've got a number of compounds that look really interesting with respect to their in vitro and in vivo properties using the methodology that I've just described, and we then go on to think about how we're going to take these compounds forward in terms of projecting human PK and dose. And the PK tool developed by Xenologic has been extremely helpful in that regard. So what we've generally done is generate rat IV oral PK data to allow us to do that human PK and dose prediction. And then at the same time, we've generated some off-target activity profile at Serup, and then we've tried to use both of these 2 things together to see what the quality of the compounds is, both with respect to human dose, and does it align with DNDi's target candidate and target product profile, and what do we think of that is our potential therapeutic index over, excuse me, one particular off-target activity which we've designated target X for this presentation. One of the advantages I think of the human PK tool is that you can change the parameters to understand to change what you think the absorption rate of a compound might be, and we've deliberately set that to have rapid absorptions right now to give us the highest likely Cmax, because we think that any activity or off-target activity with relate to this particular target is going to be Cmax driven. And then what we've done is do a bunch of modeling to try and look at what happens when we increase the dose in human until that off-target IC50 is either just reached, and then actually what happens if we're hitting that particular target for half the dosing interval, and what kind of safety window might we expect. So a screenshot now of what the tool looks like and how we've used it and just where we've ended up inputting data. So this is purely looking at rat in vivo data. There are multiple other tabs and ways to input data if you don't have rat in vivo data, for example. But what we've done is input our rat IV PK data here. You need to be a little bit careful about the units. Certainly, as a med chemist, I'm used to looking at volume in liters per kg rather than mils per kg, so I'm forever making a mistake in terms of the volume, so just need to be a little bit careful there. This Ka value is where you can modulate your rate of absorption, body weight of the animal, fraction unbound, where 1 is 100%, and then all bioavailability. And then down here, what you can also do is define what your human plasma protein binding is, and also if you consider based on clearance that actually the human bioavailability might be higher, you can tweak these figures accordingly. And that's what we've done here. We've inputted down here what our key off-target activity is. So we've put in 300 nanogram per mil, which generates this red line here. And then you can set your dosing regime, but we've set it for every 12 hours for a 5-day dosing regime because we think this is relevant to what we'll end up dosing in the clinic. And then by modulating CF, we can then dial in the appropriate AUC. So for this particular program, we think that AUC is the most relevant marker for activity. We're not looking at Cmax or Cmin. We're looking at AUC, so we can use the CF value to dial in the AUCs that we've seen in the preclinical models to get that to the right point, and then we can understand what our human predicted dose would be and also what our peak to trough looks like. And if we carry out a bunch of those different calculations, this is what we get to for this particular compound. Out of the PK tool, we generate a clearance prediction. From that, we can calculate what the unbound clearance is based on its plasma protein binding, half-life, and bioavailability. And as you can see, what we've been able to do quite easily is just modulate the dose. So this is what we think is at the top is going to be our therapeutically effective dose at 5 mg per kg, and we're well away from our target X IC50 at that point. But as we start to increase the dose up to 30 mg per kg, we might be starting to have a Cmax-driven effect against that target, and then up to 75 mg per kg, we'll be hitting that target pretty hard for half the dosing interval. So we can start making some judgments as to what we think the therapeutic index of this particular compound might be, and also we've been now been using this type of analysis to try and help inform what the de-risking strategy might be with respect to an in vivo study, for example, to look at this particular off-target activity. So just to summarize, I know I've gone through this pretty quickly, so if anyone's got any questions afterwards, then I'll be very happy to answer them. A couple of key messages to take home for this particular series and another one. We've really worked hard to try and understand the PK/PD relationship, and so far we've got to the point where we think that free exposure is a good surrogate for what's going on, and if we can hit free exposure in our in vivo model and then subsequently in human at around the in vitro IC50 for a significant period, we think that's a good predictor for high levels of efficacy. Something that we didn't know before, but we've measured and shown that actually binding in plasma, in this case hamster plasma, or any other plasma protein preparation from human, rat, dog, etc., is significantly higher than binding in the media used for the in vitro assay. So those two are not the same. And then using this Microsoft Excel tool, that's really helped us in the conjunction with all of this other data to help think about what our exposure over that entire dosing period might be, and then decide what the compounds are to take forward into those resource intensive in vivo studies. Now this tool at the moment is not that user friendly. It's very much fit for purpose for us, but if people out there are interested in that, then we'd certainly be happy to spend a little bit of time making that a bit more user friendly, and then we could send that to Mark and we could post it out for people to have a look at and use in their own projects. And then secondly, just a summary around the PK tool itself. We found that extremely useful actually, just using it on a day-to-day basis to think about whether the series or particular compounds can deliver the profile that we want. It's helped us model our therapeutic index over off-target effects. I think one thing I would say is that we've had to go to Excel to do some of those modeling of all PK data. If there's a way of being able to input that kind of data into the PK tool to do all of this in one place, I think that will be particularly helpful. Currently, you can put ID data in and it will model all data, but I couldn't see a way of inputting the kind of PK data that we generate typically into this. And obviously, I've only scratched the surface really today of some of the features that the PK tool has. Absolutely thoroughly recommend you playing with that if you've got in vitro data or different types of in vivo data, and see what it's telling you on your project. So I'm going to wrap up there, and happy to take any questions now.
Oh, crap.
Ah, Mark, hooray. So yeah, happy to take any questions, or happy to pass it back to you, Mark.
Thanks very much indeed, Gav. I'm trying to find a way of unmuting everybody, but I'm not sure I can.
Okay. Yeah, you muted me for a second.
Yes, I realise now. Sorry. I thought this was like GoToMeeting, and it seems somewhat different. There is a way of asking questions down the bottom of the tablet that you have, I believe. So if I fail to find a way to unmute everybody, then that is an option. There must be a way to do this, but I apologise. I'm going to struggle. In the interests of time, I'm going to suggest we move on. Gav, I'll make sure that if you're happy, I'll circulate your email address in a follow-up email if people have questions that they don't manage to enter into the webinar tool, and if I don't manage to unmute it.
Yeah, that's absolutely fine, Mark. But yeah, very happy for you to do that.
Okay, thank you. So I'm going to suggest we move on to Isabelle, and as Isabelle's presentation is mostly about DataWarrior, I'd be interested to know, we'd both be interested to know, have you used it before? So if you could quickly fill in your answers to that, that would be fab. Okay, about 80% of you have voted so far, I think. Okay, that's great. So just so that you can see, about half of you. I would thoroughly recommend it. I think as a free tool, well, as any tool, I think it's extremely versatile, and I hope you will agree once you've seen Isabelle's presentation. Isabelle, so I think you'll need to share your slide. You should be unmuted now. Yeah. Need to show
Can you try again?
You need to show your screen. Yeah, we can hear you. We just need to show your screen.
Okay, just a second.
That's it.
Okay, you can see my screen now?
Yep, thank you.
Yeah, okay, great. So, well, good afternoon, everyone. So now I know that you can hear me. That's great. I must say I'm quite unfamiliar with the tool, with the web GoToWebinar system, so I hope I'm going to do that. Okay. So my name is Isabelle Giraud. I'm working in the team of Thomas Sander. Thomas Sander is the developer of DataWarrior, and our company is Actelion. So it's a biopharmaceutical company which is located in Basel. So it's very next to the border, to the French border and the German border. And so if you think I'm speaking English with a French-German accent, that's probably normal. If you haven't done yet so, you can download DataWarrior on this web page. It's an open molecule website. Oops, sorry, this was not intended. Okay, so I'm going to give a demonstration of DataWarrior, and I will base this demonstration on the tutorial of Karin Lowe. She has published an excellent tutorial that you can find on this web page, so on the MMV page. And the tutorial is about using ligand efficiency metrics to optimize CCR5 antagonist with DataWarrior. And she has based the tutorial upon the analysis of 4 papers that are listed there. And she explains in the first part how to extract all the structures and their biological results from the papers using the Campbell website. And I'm going to explain how to extract these structures directly from DataWarrior, because DataWarrior has a built-in functionality that allows to connect to the Campbell database directly. So when you retrieve the data, it will look a little bit. It will looks like that. You have the structures in a column, then the reference to the paper, and the PubMed IDs on the right. And probably it's you won't be able to remember the PubMed ID which relates to the origin. So if you would like to know that this PubMed ID relates to Merck, you will be happy to merge this information into your file. And this is also a function that exists directly into DataWarrior. You just need to have a separate file which lists the origin and the PubMed ID on the right, and you can very easily attach the information. And this is something that I'm going to show you. Then I will take the data from Merck and show you how to create an SRR using DataWarrior functionality. So basically, you have to draw the scaffold or the core fragment, and then DataWarrior will be extracting the substituent automatically. And it's also very nice to be able to rank the substituents according to the effect they produce on average. So how much they can influence the effect of the compounds. Then we are going to create a view, a 2D view, where you can represent on one axis, on the x-axis, all the R3 substituents, for example, here, and other agents, so the two exit vector, and then we have another exit vector which is R2. That we will represent using the shape of the different shape for the marker. On top of that, we map the activity of the compounds on the color. So green markers mean that the compound was very active, and red means that the compounds were not so active. And what is also nice, I find, is that with this representation, you can also identify what I call holes. So compounds that have not been synthesized and that are in a good area. So we could expect that these compounds, if we synthesize them, they would have a good activity. And last but very important remark, I'm not a chemist from training, so if such a sentence means something, and if a structure pops directly into your brain, and maybe the compound's name, I can ensure you that it's not going to be the case for me. So that's just a warning for the questions part. Please be patient with me. So that's all for the slides. I'm going to go directly into the software. So I have installed DataWarrior on my Macintosh, so it's a Mac. So the user interface is maybe a little bit different for you. I launched the software. And as you can see, at the beginning, you have an empty window. And there is many different ways to get data into DataWarrior. The most obvious one is probably to go under File and then open an existing file. You may also want to have a look on existing files that are example files that are located either in reference files, and these are files for chemists or for biologists. Or in example files, you have all kinds of data. Another way is to copy-paste data, and I will show you that with this table, which is actually the table you can find into Caroline's tutorial. So it's this table that I have just copied and pasted. No, I cannot copy everything because of webinar panel. So by just copying and pasting that into an Excel sheet or OpenOffice sheet. So now I'm going to pick this, and I will go on DataWarrior and simply make edit paste. And you see that it has created a new table with the content of the OpenOffice Calc sheet, and also some additional views and some filters. Yeah. But what is more interesting for us is, of course, to retrieve the data from this Campbell database. So the menu point is here in database search Campbell database. And you see immediately that here you have the whole list of the targets that you can find also on the Campbell website. If you select one target, you will see the target details, and this corresponds actually to what, sorry, that's not, to the information you would get into the target report card. It's not as complete as the target report card, but it's almost, it takes the main fields here. So, for example, the preferred name is there, and the protein target classification is actually what you will find here. Also, you can use this hierarchical protein family filter and filter out the targets that are not related to this protein family. You can filter by name, so if you search by for Plasmodium, for example, so if I would like to retrieve all data for Plasmodium, I would select all the targets and just press on OK, but that would be a huge amount of data. What I'm going to show you for this presentation is how to retrieve the structures and the biological results that were cited or described into the 4 articles we have mentioned before. So I just need to have the PubMed IDs. So I go back to the OpenOffice Calc file. I copy the PubMed IDs, go back there and just paste it. So it's just separated by spaces. That's not a problem. Now I already trained my retriever and say I only want the data that relates to this target. But in fact, I would like to show you afterwards how to use the filter. So I will simply get all the data that have been cited in the articles, not only the one for the CCR5 target. So then I simply click on OK. And I'm left with more than 320 rows. You see that there is the structure of the compounds that have been drawn into in these articles, in these papers. The mole rate number is actually a unique identifier for the structure. Then there is the type of the assay and with the value which has been measured and the unit. A little bit further, you can see the target name. If you click on the header, it sorts the data. And you see also the PubMed IDs here. And there is this small this small one in a square. It means that there is more information into this cell. So if you right-click, you get you have the possibility to open a web page, which will display the abstract for this paper, for the paper of this PubMed ID. Also on the right, you have all the filters. So every column almost has got a filter. For example, if we want to get rid of all the data that are not related to an IC50 test, because these are the data we are interested in, IC50, we can use a category filter, and maybe it's already has been created. So I'm looking on the right, and yes, there is a category filter which has been created here, name type, and So if I display now the AUC data, you see that if I uncheck the checkbox here, the AUC data disappear, and so on. I just want to keep the IC50. It's maybe easier to just right-click in the middle of the filter, deselect everything, and then I select IC50. So if you go on the bottom part of the window, you see that there is only 138 visible records now on a total of 320. And I can really simply delete the invisible rows, the ones that are not IC50 data. So I will be deleting the invisible rows like this with data delete rows. Okay, now we will check whether the target is the good one. So we already see that there are more than just the CCR5 target. If I sort, I can see that. Is there a filter which already contains target name? Yes, there is one, but it's a text filter. So it means that you can type the beginning, for example, of the target name you are looking for, or just part of the target name. Yeah, I can also explain these small icons. This means that you will see exactly the inverse, so the one that do not contain chemo. So if I click that, you see the chemo records disappear. This is to disable the filter, so I will get again the 138 compounds. And this is to delete the filter. So now it's gone, and I don't know how to recreate it. So in fact, to recreate the filter, it's easy. It's just by right-clicking here on the header of the column. You right-click and just select new text filter, or I wanted to go into the target name, sorry. So on target name, I do the same, new text filter or new category filter. And I will do the new category filter so I can really see what are all the target name I have in this file. Again, I unselect everything. I will only keep the one that relates to the CCR5 target. And now I'm left with 91 compounds, not compounds, but rows. And then I delete again all the invisible rows. And what I'm going to do now is to append the origin of the paper next to the PubMed ID. So for doing that, I will use this table that we have prepared before. So the one that I've just copy pasted from the paper from the tutorial of Caroline. We need to save this file somewhere locally. So I will go into File, Save, and go to my desktop, and I select. I will overwrite this file. Okay. I go back to the original data set, and I'm going to merge the table into this file. So now the software asks me to designate the merge key. So now it has tried to assign reference to reference, but it's not the correct one. I want to use PubMed ID as a reference key, of course, as a merge key. And in fact, reference, I can trash it. I already have it into my file. And originator is the column I want to create, the one that contains the origin of the papers. This I can remove, it's not necessary. So you see that it has mapped the name of the origin of the paper onto the PubMed ID, and this is much more recognizable for normal users. And you can use this information also to color the content of the column. So, for example, I can color the structure according to their origin. I would right-click onto the structure header and use set background color and say that I want to color the structure according to the origin. So only we have 4 different colors that proposed. So on the very left also you have this so-called detail view, which shows you what is under your cursor, so under the mouse cursor. So you see that you don't see everything. Yeah. That the yellow are from Pfizer, the green are from Merck, and we could continue like this. The red are from GlaxoSmith, and the bluish are from AstraZeneca. Okay, now I would like to show you how to create an entire data from Merck. And I wanted to show you something else. If I sort the column value, you see that some of the records have a value which is 0. And I would like to represent the value of my compounds on a 2D view. So I create a 2D view by just right-clicking on the header of any view. I will just select value, and here I can, for example, take the structure. And since IC50s are concentrations values, I would like to have that that scale. I would like to represent that with a logarithmic scale. So I would like to select data, treat logarithmically, and use value as a logarithmic scale. But it's not possible, as you see, it's not in the list of the available column. So why is that? It's simply because some of the records have this value 0, which is probably an error when they have been registered. So we have to get rid of them before using this logarithmic scale. For this, just go into data and delete rows, the selected rows. Okay, now it should be possible to treat this data logarithmically. Yes, now value has appeared in the list. And as you can see, now the scale, adapted, the axis is now representing logarithmic scale. Now, if we want to create, to display an SRR only for the compounds that come from Merck, I have to select these records first. Here again, I can use a filter. So again, right-clicking, new category filter. The filter appears always at the end of the already present filter, so you have to scroll down till the end. I deselect everything, and I will just select the Merck compound. And now there is another possibility. Instead of deleting the rows as I've done before, I can simply extract them. So I just select all the records I would like to extract. So in this case, everything from Merck, and then I go into File, and then New from Selection. And it's also very simple with the shortcut key Ctrl-Shift-N. And you see what happens. It has just extracted the 29 records, and all the other were left.
Isabelle, I'm wondering if this is a good time to stop. We are pretty much at the hour. I apologize. I think we've had some issues with the technology. I know we talked about having a follow-up session with some additional functionality, because I know that there's some really interesting functionality from DataWarrior.
Okay.
Would you mind if we call it a halt there and add some of the things? I know you were going to show us some lovely things with SAR analysis and things. Would you mind maybe coming back another time? I'm just
Well, no, not at all.
Is that all right? Thank you very much. I do apologise for my incompetence with the technology. I should say a very brief opportunity for people to ask questions. What you do have to do is, if you use the raise your hand function, I can see if you've raised your hand and you have any points. I haven't found a way to unmute everybody, but I can unmute individuals. Okay, well, that's great. So we'll all get an update. You'll get a recording and information about future meetings. Apologise that I managed the time badly, but I would like to say a huge thank you to Gav and to Isabelle, and also to Rich Elliott, who's kind of encouraged us to make this happen, and of course to all of you for coming along. I hope you found it useful. Please let us know, and I hope to see you all again on the 25th of February. Thank you very much indeed, and thanks again to Isabelle and Gav.
Thank you.